Question number 4. Individuals with sickle cell disease have a form of hemoglobin called hemoglobin S, which is less efficient at carrying oxygen. The diagram shows how a mutation in the gene coding for hemoglobin causes the production of hemoglobin S. So, our first job, sickle cell disease is caused by a substitution mutation. Describe evidence from the diagram that supports this statement. Okay, so let's take a look here. We've got our DNA sequence here for hemoglobin, our normal hemoglobin, and our hemoglobin S. Pause it for a second and see if you can see what the difference is here. Where is there a discrepancy? If you look right here and here, those two bases are not the same. Okay, so we've got GGC, CTC, and here we have GGC, CAC. So we've got a mutation taking place there. If we look after the point of mutation, we don't see any sort of like frame shift. That after the point of the mutation is totally fine. TCTC, CCTC. If we look at the amino acids we produced after the point of mutation, it's the same. It's just that nucleotide that's changed, just that base. So therefore, because it's just that base, we know this is a substitution mutation. So what's the answer here? You could say a thymine is swapped with an adenine. You could also say a T is swapped with an A in the mark scheme. That's totally okay as well. But that's how we know there's a substitution mutation taking place. You could also say, oh, that there's a substitution mutation because the sequence in amino acids is different, and because the second or one codon in this whole sequence has a different base or a different nucleotide in it. So suggest how changing one amino acid could cause the hemoglobin S protein to function less effectively than the normal hemoglobin. So if we've changed one of the amino acids, and this is not specifically about hemoglobin, but just generally, if you remember, if we're changing an amino acid, what's going to happen to our protein? Lots of different things, potentially. So for this answer, we could say that the shape or the structure of the protein has changed. Okay, so the shape and the structure is not going to be the same. Or we could say that oxygen will have a different binding site. I'll just move my face there so you can see that. Oxygen might have a different binding site or might bind differently. You could also say that the protein doesn't fold properly, or that you're going to get different bonding between the amino acids. Okay, and that's all about the shape and the structure after we've kind of done that translation and how that protein's going to fold so it can function properly. So the whole idea here is that if you change one of the amino acids, you're going to change the structure or function of this protein. So B, compare the effects of frame shift and missense mutations on the resulting sequence of an amino acids in a protein. So frame shift and missense mutations. These are words that we talk about when we talk about mutations of proteins. Let's take a second to remind ourselves what they mean in an example we can always bring to mind to help remind us. So to illustrate the different types of mutations I've got 3 different sentences here, and we're going to talk through them. So the first one here is my normal sentence. The cat sat on the mat. Okay? A missense mutation would be a mutation that causes a change to this sentence, okay? The cat sat on the bat, okay? So that's not the same as the original. The original is mat. Now that M has changed to a B. So there has been a change to my sentence. However, it's still a sentence that you can read in English and it kind of makes sense as a sentence. The meaning is not the same, but the sentence is still a sentence. That cat sat on the bat. I don't know why a cat would sit on a bat, but it did in this sentence. So even though that's not the original meaning we were trying to get out of it, it still makes sense as a sentence. And that's caused because we've got one change taking place in our sentence that's changing kind of the meaning of the sentence. A frame shift mutation will cause an absolute mess, okay? So here in this frame shift mutation, I've deleted my letter A here. So instead of saying the cat sat on the mat, which is the original, I'm saying the don't have an A. And so I'm having to compensate and change all the letters after the point of that change, and it's reconfiguring my whole sentence, okay? So I did have the cat sat on the mat. Now I have the ato in mata. Makes no sense. What am I trying to say here? A missense mutation, okay, is going to cause a change only to one amino acid in our whole sequence. So in this example, only one letter in our sentence is changed. It will change the meaning of the sentence a little bit, but it's still a sentence, okay? If we're thinking about amino acids and proteins, it's kind of like saying you change an amino acid, your protein will be different. It won't work maybe as it's supposed to. It might be slightly different in function, but might still have a bit of chance of, functioning in a way that's somewhat useful. Frame shift mutations cause an absolute mess because in a frame shift mutation, the order of amino acids after the point of the mutation are going to change. The same way the letters after the point of the mutation in a sentence have changed, and that just changes everything. Question C. An individual with sickle cell disease has the homozygous genotype SS. All their hemoglobin is type S. Their red blood cells can develop a distorted sickle shape and are stickier than normal blood cells. An individual with sickle cell trait has the heterozygous genotype HS. So their red blood cells contain both forms of hemoglobin because they've got the normal gene and they've got the disease-causing variant. So explain why the inheritance of sickle cell trait is an example of incomplete dominance. So let's talk about incomplete dominance for a second. So when we're talking about genotypes, we usually have two distinct genotypes that we're discussing. So for example, in this example here, we have a red rose and a white rose. And these would be our two very distinct genotypes. We're usually used to thinking that whatever has the dominant allele will express that dominant trait above a recessive trait. Incomplete dominance says that instead of you expressing one specific trait above another, you kind of express both. So in this example, we have a red flower and a white flower. Because the red is big R, we're kind of used to thinking, oh, that's probably going to be the dominant trait, so the next generation should be a red rose. But actually, we're going to kind of express both genotypes. So we're basically going to say it's kind of red, it's kind of white, therefore it's pink. So you kind of end up creating a third genotype and phenotype that is kind of unique. Similar thing will happen with sickle cell. The sickle cell disease is going to be its own thing with two disease-causing alleles. The sickle cell trait is heterozygous, so one normal allele and one sickle cell causing allele. So instead of it causing a full-on sickle cell disease, it's going to cause something called sickle cell trait, which is its own thing because you're going to express both of those alleles. You can still produce normal hemoglobin, but you'll also produce some of this kind of weird sickled hemoglobin, which is going to alter your red blood cells in their shape. So you kind of express both of those things. And that's why the answer to this question is that the reason that this is an example of incomplete dominance is because they can express both types of hemoglobin, the normal type and the sickle type. And it's almost creating a third genotype and phenotype, which is its own unique thing. Question II suggests why individuals with sickle cell disease have an increased risk of stroke. Well, they've told us in this question that when you have sickled hemoglobin or type S hemoglobin, it makes your red blood cells stickier than normal. That's a problem for forming clots. So the reason people with sickle cell disease have an increased risk of stroke is because you have an increased risk of your blood cells sticking together or sticking to the walls of your arteries and creating blockages. Or you could say that, these red cells will stick together and block the arteries to your brain, which is what causes a stroke. So that's the reason why individuals with sickle cell disease have an increased risk of stroke. So finally, when the population of the UK was 67,620,000, one in every 4,600 people had sickle cell disease. So we're supposed to calculate how many people had sickle cell disease. Okay, if we figure out how many people there were, so there was one in every 4,600 people, and we knew that there was a total of 67,620,000 people, it means that if we times those two things together, 1 over 4,600 times by 67,620,000, that would tell us that when the population was this size, how many of those people had sickle cell disease? And the answer is 14,700.